Good afternoon, everyone. Thank you so much for joining us. My name is Katie Rowley. I'm with the NOAA Central Library, and we are very excited to bring another installment of the NOAA Innovator Series to you today. First up, a few logistics. We are accepting questions during this presentation, so please place those in the question/chat panel, and we will be holding those questions until the end of the presentation. Also, if you are having any trouble with the audio or visual components of this webinar, please try logging off and logging back on. If you're still having any troubles, you can chat me or you can email library.brownbag@noaa.gov and I will try and help you troubleshoot that. We are very excited that this partnership has continued with the Technology Transfer Office. And with that, I'm going to turn it over to Derek Parks to introduce our speaker.
Thank you very much, Katie. Good afternoon, everybody. I'm glad you're all able to join us today for our next installment of the NOAA Innovators Brown Bag series. As Katie said, this is Derek Parks, and I'm the Acting Deputy Director and Technology Transfer Program Manager for NOAA's Technology Partnerships Office. Our office with the Central Library has teamed up to organize this series to highlight our innovative technologies that are being developed by NOAA scientists and engineers, as well as technologies developed through the NOAA Small Business Innovation Research, or SBIR program. So today, in that vein, we will hear from Dr. Jason Kriesel of OptoKnowledge Systems. OptoKnowledge Systems has developed deep ocean sensors that can help determine the role of methane in the global carbon cycle. The device they developed is designed to be deployed on an ROV and can stream methane concentration and isotope ratio measurements to a host ship in real time, which is pretty cool. OptoKnowledge received an initial SBIR grant from NOAA in 2017. And so for those of you who don't know, SBIR provides a three-phase competition-based program for small companies to develop research, to conduct research and development on behalf of NOAA and transform their innovative ideas into commercial products. So all the SBIR proposals must directly benefit the NOAA mission and have excellent commercial potential as well. The technology Jason is presenting today is currently in the commercialization phase of SBIR after they received funding from NOAA's Office of Ocean Exploration and Research, which is also fantastic. And as an FYI for anybody out there, from other federal agencies under federal contracting rules, SBIR developed technologies can be acquired through a sole source procurement mechanism by any federal agency, which is a great benefit of the SBIR program. And now just a brief background on Dr. Kriesel. He is the lead scientist at OptoKnowledge, leading the development of the sensor technology, for a range of applications. And he has a PhD in physics from UC San Diego. And he was also an NRC postdoc at the National Institutes for Standards and Technology in Boulder, Colorado. So it is my pleasure to introduce to you Dr. Kriesel. Take it away.
Thanks, Derek. Thanks a lot for this opportunity. I appreciate the chance to talk to everyone about this technology. As Derek mentioned, the funding for this work came through the SBIR program initially. Now we're in what we'd call a phase 3 with additional NOAA funding to raise the technology readiness level, test and deploy it. And for the SBIR work, Margot Bowen was our technical advisor, and Chris Breverson was involved, and now he's very much involved in the phase 3 effort that's ongoing. And I want to acknowledge my collaborators now, and then later I'll talk about the specific roles each collaborator is playing. We have scientists from the Jet Propulsion Laboratory, JPL, PMEL in Washington, along with University of Washington and Oregon State, all NOAA affiliates there. And OptoKnowledge, we're a small business in Torrance, California, just outside of Los Angeles. So this is a totally West Coast collaboration. So I'd say good morning to everyone on the West Coast too. So let me begin by providing motivation. So, methane important for the carbon cycle, and it's very nuanced. So there's still a lot of questions about methane's role in the carbon cycle, in particular when it comes to the oceans. So, you know, various sources have been discovered recently, many seeps off the coast of Washington and Oregon, hydrothermal vent sites, hydrates, all these potential sources of methane, and there's still work that needs to be done to understand the fate of that methane in the water column. Does it make it to the atmosphere? And in situ sensors would help answer some of those questions. In particular, isotope ratio sensors could help attribute different sources. So on here on the right, there's a diagram showing the range of isotope ratios. So, you know, typically methane, carbon 12, and 4 hydrogens. Most of the methane is that standard isotopolog. But there are naturally occurring stable isotopes with a carbon 13, about 1% of the methane, and also stable isotopes with one of the hydrogens being a deuterium. So if you look at sort of a shift, a small change in those ratios, you can attribute different sources of methane to different processes, whether it be from a thermogenic source or from bacterial sources. So, you know, this type of information, having an in situ methane sensor that can do, in this case, both concentration of methane and isotope ratios, can help find more seeps and better characterize them. Look at some of the interesting biology processes going on at some of these vent sites. Also understanding the fate of the methane. You can look at some of these micro communities and better study the processes going on, and also be able to do some interesting studies in situ. There's a industry need as well in terms of energy sources, potentially identifying leaks in production facilities, you know, being able to attribute a leak versus just a background of what's already there is an important application. So there's the NOAA need is really what we're focused on in the current phase of this work. But with the SBIR, you know, we do have a commercial application that is being pursued simultaneously. So moving on, the sensor I'm going to talk about is a laser absorption spectroscopy sensor. So some of the pioneering, I'd say, work done doing in situ methane measurements with a laser-based sensor was about 10 years ago. A system put together by Los Gatos Research and deployed by Harvard used one method of laser absorption spectroscopy, and we're doing a different method, and I'll talk about the differences in the next slide. But this was a successful, I'd say very successful project of demonstrating the ability to do such laser measurements in situ at depth. So this system used an ICOS cavity, which I'll talk about. And one of the things that differentiates this prior system to what we're doing is that this prior system used a membrane. So that's a standard way of doing the sampling. But for isotope ratios, it can cause measurement problems. It does fractionate, so it can change your ratio. You know, from say methane with carbon 12 versus methane with carbon 13, they react differently on the membrane, and that can bias and skew your results. And that fractionation is temperature dependent, and it's very hard to calibrate. And it's just sort of if you could get rid of the membrane, these measurements could be more precise and accurate. And that's exactly what we're doing with our new concept. The system, was able to do concentration measurements. It required fairly high concentrations, in particular to do the isotopic measurements. But it was deployed on an ROV, shown in the pictures, and was able to get some good measurements. So going on to the difference between, this prior system, it's an integrated cavity output spectroscopy system. So in this concept, you use a membrane, you get a gas sample, and you put it into a gas cell. That's what's shown on the left here. So with this concept, you shine a laser beam in. It's got highly reflective mirrors. The beam bounces back and forth many, many times. You get absorption from the gas inside, and you have a signal that's it's an integrative signal because you have a leaky mirror on the outside. So in effect, these types of systems are good at getting a long path length, which gives you more absorbance, and that gives you higher sensitivity. This type of technique is well proven. It's one of the standards for doing isotope measurements. There's, in addition to Los Gatos Research, there's a company called Picaro that makes systems for isotopic measurements, and there's some of the standards for doing isotope measurements. Although putting them in the field is often a challenge, and in particular putting them underwater at depth is an even greater challenge. So the difference between this type of concept and the concept that we're developing, the concept we're developing is called a capillary absorption spectrometer. It uses the laser. You have gas in a cell. The laser interacts with that gas, and you have absorption that you measure. So the difference is our cell is a hollow fiber, which is basically a capillary tube made of plastic or glass that's coated on the inside with a mirror. That mirror guides the light from your laser to your detector, and it also holds the sample, and you get this near unity overlap between your probe laser and your gas under measurement. And the key difference is really the sample volume for these types of concepts. Basically a mirrored straw has an internal volume on the order of a milliliter, whereas these ICOS cells are closer to 100 milliliters. So with this smaller sample volume, we're able to get away from a membrane. We're able to do discrete sampling of a small little slug of water, on the order of a milliliter itself, degas that water, take the gas out of that water, and put it into our gas cell. One other kind of thing I want to point out, which I'll touch on in a slide or two, is that with this concept, we're using mid-infrared wavelengths. And those are much stronger features than what's typically used in an ICOS, which is in the near infrared. So the near infrared is used because you can get very good mirrors. You can get these high reflectivity mirrors in the near infrared, but you do with the mid infrared, you have better access to fundamental spectral features, which have higher sensitivity. It's also got a simplified spectral analysis. It's a direct absorption technique where you're looking at just a direct signal on your detector. So I'm going to come back to the capillary absorption spectrometer and talk a little bit more about this technology, because that's really what OptoKnowledge is developing, and what was supported by the SBIR, and it's the concept that we're also pushing to commercialize. But before that, I want to give an overview of the system and talk just a little bit about the sampling, and then I'll come back to the infrared spectroscopy and that CAS system, that capillary absorption spectrometer. So a very simple diagram of the overall concept. We sample ocean water that has some dissolved gas in it. Take a little discrete slug of water on the order of a milliliter. We degas that. The gas goes to our hollow fiber spectrometer. We measure, the laser goes through the hollow fiber, the gas is inside, and we have a absorption signal. So there'll be features, in this case, light is absorbing at a specific wavelength corresponding to methane with a carbon-13, and there's different features with methane with a carbon-12. And so you can, from these different signals, you can get an isotopic ratio, and you can also get the concentration. So on the sampling, just a brief mention of the sampling. This was developed by Max Coleman, the concept at JPL. First demonstrated under the SBIR. We've been iterating on it, continue to iterate it on the current project. So, again, the idea is you take in a discrete sample and you degas it. So the front line of this water at depth is a valve that's rated to 5,000 psi. That's basically the only part of the system that's in direct contact with the high pressure water. And then after that, it's, we take a gas sample and put it into the CAS. So it's a new concept. I think it's part of the innovative work that's been going on in this project. We've demonstrated it works. It's very, we're now getting reproducible results. We can, infuse methane in water and get, gas concentrations and isotope ratios out. So I'd say, that's all the good news. I'd say the downside, which may be obvious, is that we have a water sample. We have old wastewater that we're continually collecting throughout a measurement sequence. So they're small little slugs. In our current design, we're taking 0.5 milliliter at a time, but we are storing all that on board. We're not trying to push it back out. So eventually we're going to accumulate water over time. So, we can get 1,000 measurements and have 0.5 liter, which is kind of typical. It's kind of what we're aiming for right now. But that is, a limiting factor. If you wanted to have a long duration, you wanted to have the system down at depths for a long time, there needs to be some other way, I think, to get rid of the water. For now, the plan is we'll make 1,000 measurements or so. The system will come back up. We'll have to dump the water. So moving on from the water sampling, I'm going to go back to the spectroscopy, so the gas analysis. So the concept for the gas analysis is based on laser absorption spectroscopy. So, and with this technique, you can uniquely identify molecules by their spectral signature. And they are, this is called the molecular fingerprint, and you can it's very good at identifying, but also quantifying. So the spectral pattern tells you what molecule it is, your absorption, and the degree of absorption, you're able to quantify it very accurately. So, I've shown some example spectra here on the top right for different species. So methane has, very prominent features in, beyond 3 microns. So this is, down here at the left-hand side, this is more the near infrared region. 1 to say, gets a little gray, but 2 microns and then 3 microns and above is what's typically called the mid-infrared, the longer wavelength. And the methane has a strong absorption, water strong absorption, CO2. This is, why they're potent greenhouse gases, because they absorb very well in the mid-infrared. But they are all distinguished. There's a bunch of different spectral features corresponding to different rotational and vibrational modes of these molecules. The plot on below just sort of gives you an overview of where different species absorb as far as wavelength. Again, down here, visible to near infrared, and then mid-infrared going on. And so one thing I wanted to kind of come back to the plot on the top right, the mid-infrared is stronger absorption features. The near-infrared, typically used for ICOS, much weaker features. And again, the reason they use this region is because you can get much better mirrors in that region. There are work to do ICOS and mid-infrared. It's just more difficult in terms of the optics and things like that. So so laser spectroscopy specifically for methane isotopes. So if I go back to this just for a second, there's this cluster of features for methane around 3.2, 3.3 microns in this particular case. If we kind of zoom in a little bit more on that region, there's similar but different features for methane and carbon-12 and methane with carbon-13. And if you blow up a specific region even further, you can find spectral features for methane with carbon-12 and methane with carbon-13 that are close to each other. And this is one region you can exploit to do isotope measurements of methane with laser absorption spectroscopy. So there's also water. These are very weak water lines actually, and they're well separated. So here up in the top right is a actual measurement, and the red line here is what would be called a baseline measurement, where it's just the laser scanning with no gas. So you scan the laser in wavelength, that's the x-axis. You have a detector signal. It varies because actually the power of the laser changes as you change its wavelength, but that's very stable, very fixed. And then with gas introduced, you have this blue curve where you have little absorption features at specific wavelengths. If you convert this detector signal, so you have a loss to the absorption, if you convert it into absorbance, which is the typical way you would view this type of data and compare it to a database, you can see the stronger absorbance here for carbon-12, specific pattern, and little features for methane with carbon-13. So again, this is the way you do the measurement. You have these spectral features that are unique to the different molecules. In this case, there's even unique features to the different isotopologues. They're distinguishable. You can uniquely identify it, and you can quantify the ratio of the two isotopologues. So to the capillary absorption spectrometer, back to that. As I mentioned, we have a laser coupled into a coiled hollow fiber. We coil it up. That hollow fiber is a tube that we deposit a mirror on the inside. So it's a dielectric mirror. There's a silver layer, and we put a dielectric layer on the top of that. So we make a very reflective mirror specifically tuned for these mid-infrared wavelengths that we're working with. So it's the light is guided very effectively from one end to the other. And then the gas is also introduced into this hollow tube. So like I mentioned before, basically it's like a mirrored straw, and typical lengths are about 5 meters. So we'll have about a 5 meter hollow fiber that will coil up, and that's our path length that the laser interacts with the gas. So we can change the diameter of the hollow fiber. I mean, we have, we just use a different glass tube or plastic tube with a different diameter, and that changes our sample volume. There's a trade-off there because, if you want to go smaller sample volume, then you'll have longer throughput times to pump the gas all the way through the system. And you'll have lower transmission of light. So there's this trade-off here. Typically, we're using a larger diameter fiber to get faster throughput. So the gas can be pumped through the system. This 5 meter long tube that's 1.5 millimeter diameter, we can get the gas through in about a second. And that has about a 10 milliliter volume. But again, we can vary this depending on the application. If we're very sample starved, we can go towards the smaller end. But if throughput is a priority, then we'll go to the larger end. In any case, it's compact system. You can coil it tightly, and it's relatively simple. You don't have this very sensitive alignment, and it's robust. So example systems that we've made, I'll talk about the relationship between this system and the underwater system, but we've made an airborne version with methane. Here's a picture of an actual system, a laser, coiled fiber, detector. All the electronics are below here, and there's a vacuum pump down here in our, what we call the little basement. You close the box and this whole thing is temperature stabilized. This system streams data via Wi-Fi to a host laptop that's running a LabVIEW graphical user interface. So this version has been, we've flown it on a drone, did measurements. This was just done in January. Sorry, joys of working at home. The system can measure leaks, and I'm just going to move on from there. So the overview of methane sensor development is depicted in this graph. So I want to kind of take a step back here and just sort of say where we are and kind of the relationship from the SBIR to our current project, as well as this kind of synergistic airborne project. So we've developed this fiber spectroscopy system. It started with a collaboration with Department of Energy Lab, PNNL in Washington State. We produce the fibers ourselves. It's our proprietary kind of technology. We collaborated with JPL for the original NOAA SBIR. They did the water sampling. That work is directly feeding into our current, we have a NOP project funded by OER, and that's the collaboration which includes JPL as well as PMEL, University of Washington, and Oregon State. So where we are right now is, I'll come to, we're packaging the system for use on a CTD or an ROV, and eventually we'd like to get to an AUV. There's a little gap here because that will require additional work to reduce the size, weight, and power. So kind of going on from there, it's the current project, what we're calling our phase 3, which is OER funded, not project. The goals are, we developed this technology and now we're packaging it for deployment at depth. And so the main point is to really raise the technology readiness level, demonstrate and validate this new sensor concept. So which is the capillary absorption spectrometer with this new in situ discrete water sampling without a membrane. So the sort of goals are concentrations with a picomole of methane, so that equates to about nanomolar concentration in 1 milliliter of water. Isotopic sensitivity, we need a more like micromolar type of concentration. And then there's sort of kind of two modes of operation where you're maybe just doing the concentration measurements at a faster rate. And then when you have something of interest, you grab and you can dwell on that one sample. And by integrating your signal over a longer period of time, you can improve your precision and do the isotopic ratio measurements. Isotopic precision, we want 1 per mil for the carbon-13. Onboard autonomous system control and data acquisition is just running on its own and streaming data back to the ship. A depth of 3,000 meters and a power right now for these CTD and ROV applications is around the 75 watt level. And as mentioned, for an AUV, we would definitely have to reduce that. So where we are now, we have the guts of the system, and we're currently working on packaging it in the underwater pressure housing. So here's, we're getting this housing from a company, Prevgo in Arizona, cylindrical housing. It's about, the internal dimensions are 10 inches diameter, 30 inches long. It's actually a lot larger than we need it to be. But since we were shooting for a CTD and an ROV for this initial application, we decided, to err on extra space just to make it easier for us for this initial deployment. Runs off of 24 volts, which we can get from either a battery or from ROV power. So, we've looked at deploying it on a CTD. We could either, PMEL has a CTD with a cutout that we could put this in, but also, for other CTDs, we could put it down in the bottom here. We've looked at putting it on ROVs. And as I mentioned, the data would be streamed back. It's streamed back via an Ethernet cable. In addition to our in situ measurements, we want to have other ways to, validate the system. So, in all the deployment scenarios, there'd be other ways to collect samples. So I just want to touch on one of these, the method of the gas tight sampling, which is what we plan to do when we deploy this on an ROV. And I just want to kind of touch on that because I think it points to, one part of this work where we're really going to do very careful validation and testing, just to see how well the system's performing in situ, but also point to what the current techniques are for doing these measurements. So this is a lot of work done by Tamara Bomberger at Oregon State, also in collaboration with the University of Washington and NOAA, Marv Lilly at University of Washington. So you sample, you take a sample from an ROV. That sample is processed on board the ship first, stored in some vials. So this is an involved process in itself. Then those samples are sent to a lab, and now currently it's being done in Zurich. That's where they have developed very accurate, high precision techniques to do this analysis. But it's a multi-step, labor-intensive process that it works very well, but probably not a whole lot of people can do this. I think Tamara can do it, Marv Lilly can do it, but it requires quite a bit of know-how in terms of making these accurate measurements. So the whole, kind of the point in this is that part of what we're trying to do is provide a different tool. So you can do many more measurements and you can get the information in real time. So deploying the sensor, I'm going to talk about a couple places that we've planned for and want to deploy the sensor. So axial seamount, volcano region off of the Northeast Pacific coast, off the coast of Oregon. Very interesting site to study. We had planned to participate in a cruise in September 2020, in a few months here, and we just recently pulled out, and mainly because of additional challenges due to COVID-19. There was challenges in terms of, putting the system together and doing, initial testing before we take it out on this cruise. In addition to that, it was a relatively long cruise. There was a lot of, we were piggybacking on it. There was other people piggybacking on it, and it kind of grew and grew. And then there was an additional 2-week self-isolation period before the cruise. So it ended up being, it would be a 5.5-week commitment, which we couldn't do it for this stage of the project. So, that said, I think we have a very good backup plan now. We're planning to do initial testing at Puget Sound. Again, this is, a West Coast team, and we have a lot of the science driven by David Butterfield at PMEL, University of Washington. And this would be much more convenient for doing initial testing. There is interesting methane activity at Puget Sound, and this, so we could not only just test the system, but actually get some interesting data on the first time we really throw it in the water. It's also logistically much more simple and lower cost. And so, the target ship would be the University of Washington's Rachel Carson, which is shown here. It has a CTD winch. And following this type of initial test, I think the current goal would be to look for an opportunity to measure on the Cascadia margin, which is off the Northwest coast. It's a very interesting site. PMEL and OER and Oregon State have been looking at this region, have recently, over the last few years, found more than 1,000 seep sites. So of methane. So it's, a very new region, and that was only with 1/3 of the margin surveyed, as it reports. So understanding this region, I think, could be very useful in terms of understanding what's going on with methane. And it's a lot closer than the axial seamount, and maybe there would be an opportunity to take the sensor out there next year. So that's what we're working towards. Just to touch on the phase 3 team, the active work that's going on. I just wanted to acknowledge the work of my collaborators. Andrew Farland is helping quite a bit with the hardware, working on the system development. Max Coleman, JPL, doing the water sampling. Dave Butterfield, as I mentioned, is really driving this from a NOAA perspective in terms of having a pull of the sensor technology that would be very useful for his studies. Marva Lilly, University of Washington, helping tomorrow with the sample analysis, the validation, and then tomorrow is doing a lot of work on the field work and the scientific studies of places like the Cascadia margin. Chris Beaverson is also driving this from the NOAA, driving this from the NOAA perspective. Very supportive, very grateful for his efforts on this project. So with that, I'll wrap up, summarize, and then open up for questions afterwards. So, methane's an important species, and in situ measurements would help facilitate studies. Instead of, taking a few samples and doing a labor-intensive measurement back at a lab, if you had something that was in situ that could give you measurements right away, I think it could help studies quite a bit. So we have a new laser-based gas sensor concept, and we're working to put that in situ. And one of the things that is kind of unique about this new system is that it's got a small sample volume, and that means you don't have a membrane. You don't need to use a membrane, which avoids some of the isotopic fractionation problems that membranes impose. Right now, the system's being packaged in a pressure housing. We're planning on testing it Puget Sound on a CTD in the fall. We are looking to deploy it on Cascadian Margin on ROV next year. And we're also looking on potentially doing follow-on efforts to reduce the size, weight, and power for AUV deployment. So thank you for the time and attention. I appreciate it. Derek, is open up for questions for you.
Thank you so much, Jason. This was awesome. Derek, do you want to kick off the questions?
Yeah, thank you, Katie, and thank you, Jason. That was amazing. I just love to see such an innovative design. That capillary tube design is fantastic. So congratulations to you and your team on that. My one initial question while people are typing in their questions would be regarding the water sampling unit. Have you had any trouble with fouling of that particular piece of equipment, or is it something that's easily cleaned after you've accumulated a half liter of water and after 1,000 samples?
Yeah, I think that's definitely something we need to study. I'd say at this point we haven't put it through the ringer in terms of how easily it's going to foul. It's something we're thinking about what we're going to do. But yeah, I think you bring up a good point where it's there's potential for fouling. We're going to have filters in there. Part of the sampling, one concept, we're going to use the hydrothermal fluid sampler. But I'd say at this point, we don't know how bad it's going to be.
Yeah, interesting. Very cool. I'm also really excited about the work you're doing for the air-based sampling too. That looks really cool. But I will defer my time to everybody else and see what kind of questions we have, Katie.
Awesome. Okay, our first question was asked a little earlier in the talk from Andrew Rose. Is it possible to configure this for hydrogen sulfide detection in situ as well?
Hydrogen sulfide is definitely a gas that can be measured with this technique. We have not looked at how well that comes out of solution, so I can't totally answer that question because it's just something I haven't looked at specifically. If I take the water sampling part out of the equation, then yeah, definitely hydrogen sulfide could be used with laser absorption spectroscopy. But it could be, if that's an interesting gas to look at, that's something that we can see how well the water sampling concept would work with that.
Great, thank you. Our next question is from Rushan Gao. I wonder what is the performance parameters of your UAV-based CH4 sensor?
The performance parameters. So the methane concentrations, so it all depends on how long you're integrating for. But let's just say we're doing update rates at 1 hertz, so 1 second measurement times. We'll get, about a 0.1 ppm precision on that. And we can get, depending on the concentration level, we can get close to the 1 per mil goal of isotope ratios. At the background levels of 2 parts per million in atmosphere, that would require more like a 10-second measurement time to get the 1 per mil precision. I hope that's kind of what you had in mind.
Thank you. Okay, our next question from Catherine Gregory. What is the obstacle involved in emptying the wastewater on board?
So the obstacle in situ would be trying to push it back out into this high pressure environment. The inside of the pressure housing is air. It's that atmospheric pressure essentially. So the water, when you're at depth, the high pressure water can come in easily, but trying to push it out would be a challenge. So that's basically the crux of it.
Thank you. Our next question is from Kimberly Galvez. In the long term, would it be possible to recalibrate and measure other gas species?
Definitely, yep. Definitely it would be. The spectroscopy measurement technique is applicable to a wide range of gases. The question comes to the sampling. Methane comes out, degasses very well, and so any other gas, we'd have to look at that. You can, if you're just looking at gas concentrations, you could use this with a membrane. And this system with a membrane could analyze many different types of gases, depending on, the one thing I should say is you have to pick a specific laser for each gas, because they all have different wavelength ranges. And the low power lasers typically only access 1 or 2 molecular species, because they only can access a relatively narrow wavelength range. But there are new developments where you could have more broadly tunable lasers that could access a wider range of molecules.
Thank you, Jason. Our next question comes from Steve Ruberg. We have karst features in the Great Lakes producing biogenic methane at depths of 120 to 200 meters. We have a Saab CI ROV. Is there any interest in testing the sensor in the Thunder Bay National Marine Sanctuary?
Yeah, sure. Please send me an email.
Thanks, Jason. And is your email on this last slide? Okay.
Yeah,
Yep.
it is. It's kind of hidden right here. I'm not really sure where to put it, so it's right after the last bullet.
Thank you for pointing that out. I'm out of questions here. Derek, did you want to ask a couple more?
Actually, I do have the non-scientist question of how do you do the degassing? Is that done through heat in that unit?
It's done through basically pressure. So we evacuate the sample, like the sample separation volume. It's in a vacuum. So the water comes into this evacuated space, and basically just the methane comes out of solution.
Cool.
Yeah.
I like it. All right. Well, that's all the questions that I had. Do you have any others, Katie?
We did have one pop up while you were talking from DJ Zhang. Have you tried to measure 14C-CH4 as well?
So 14C can be measured. It's just such a low relative concentration. It's extremely difficult. So it would have to be, it would be, you would have to do something to get that concentration up, because the natural abundance of 14C is just so low. It would be difficult to see it.
Thank you, Jason. I have run out of questions from the audience, and it sounded like Derek didn't have anything else to input. So I am going to give everyone back 10 minutes of their day. If you do have a question that you didn't get a chance to ask or something you think of after this presentation, please either email Jason from his email here on the last slide, or you can email library.brownbag@noaa.gov and we can pass that on for you. And with that, thank you everyone for joining us today, and I hope you all have a safe and healthy Thursday.
Thank you, Katie, and thank you, Jason, both.
Yeah, thank you.